Hi everybody, so today we are going to be talking about the retina, specifically rods and cones. Before I get started though, I'm going to do this video in two different ways. The first thing I'm going to do is we're going to talk about this and for a high school class. And then what I'm going to do is if you're watching from a high school class, I'll tell you when you can stop watching. And then I'm going to expand on it. So if you have a college anatomy class, we'll go on and continue on about what you would need to know for college anatomy. First thing is, if you notice the way I'm dressed, today at my school, it's actually Spirit Week. This is Comfy Day. Later on in the week, we're actually doing Support Your Team Day. So I am an LA Kings fan. This is a shirt I usually wear around the house. And let's go ahead and get started. So what we're looking at here is basically the back of the eye. This is going to be my retina, okay? This is the eye right here. So if we were to look, my retina is actually going to be in the back. This is where my rods and cones are. Light's going to enter through. This is the front of the eye. There's my cornea. The light's going to come back and hit the back of the eye. So let's talk about the different parts of this. I have this part here, which is basically blood vessels in the front. It might look solid, but light can pass through this pretty easy. If we look back in here, this part here, I'm going to call my pigmented layer. Okay, this is my pigmented layer right here. The pigmented layer is going to have a few different functions. One of the things the pigmented layer is going to do is going to absorb excess light. All right, so as light rays come through here, if they don't hit a rod or a cone, the pigmented layer will actually get this, will actually absorb them. I'm going to talk about the pigmented layer, the anatomy, just a little bit when we get over there. The other thing that will happen is as our rods and cones work, because they're constantly being activated, what can happen is they break down a lot. And so they have basically byproducts of being broke down. What the pigmented layer will do, these pigmented cells, they will actually phagocytize those. So they phagocytize byproducts, I'll put, of the rods and cones. And then the other thing is, I don't have it drawn here, but back in here is my choroid. And the choroid is going to contain blood vessels, and basically the nutrients and things are going to come out of the blood vessels through the pigmented cells and into the rods and cones. So they're going to provide nutrients for rods and cones. Okay, and that's basically what the pigmented layer is going to do. Now, we have our rods. The rods would be the orange, and then we have the cones, which are the ones that are cone shaped. So we're going to have rods and cones. Let me put this over here. We have rods, and we are going to have cones, okay? On the rods and cones, rods help you see at night. Cones help you see in the light. Okay, they're also going to help you see color. These are going to be, they're basically going to see black, white, and gray. Okay, so they're going to help you see black, white, and gray. These have a pigment called rhodopsin, which we're going to be talking about. These have pigments called photopsin. These we have about, of the rods, we have about 120 million rods. We have about 6 million cones. Okay, so you can see we have about 20 times more rods than cones, right? And our rods are very sensitive to light. The other thing is, I have it basically here 1 for 1. I have one rod and one bipolar cell, one rod and one bipolar cell. But for the most part, I could have up to 100 rods per bipolar cell. I'll talk about those in just a minute. So I can get 100 rods per bipolar cell. And these are usually 1 for 1. One cone per bipolar cell. Okay, so there's rods and cones, okay? Let's finish talking about this right here. If you notice, I have this brown line right here. That's going to be a horizontal cell. Now, if you notice, I have my horizontal cell drawn vertically, that's because I can't make it come out this way towards you. So just imagine this is coming out, and this is going to connect to different types of cells. And it's going to basically play a role in inhibiting impulses between these photoreceptors, the rods and cones, and the bipolar cells. So this is going to be a horizontal cell. And I'm sorry, they basically inhibit between rods and the bipolar cell. So that's my horizontal cell. These are going to be bipolar cells. Okay, there's my bipolar cells right there. They're called bipolar cells because they basically just have a dendrite and an axon. These are going to be called amacrine cells. So if you notice, my amacrine cells are between my bipolar cells and my ganglion cells, and then these are ganglion cells. Okay, those are ganglion cells. And then this here, my ganglion cells are going to come together to form something called the optic nerve. And the optic nerve is going to take impulses to the brain. So let's go over here and take a look at how all this works. We want to go right, maybe go back just a little bit to right about there. So this is my pigmented layer again. Now, if you notice in my pigmented layer, I have melanocytes. Okay, and what melanocytes do is they produce something called melanin, and melanin is going to absorb light rays. So as light rays come down, if they don't hit my rods or don't hit my cones, they're going to be absorbed by this pigmented layer. These are nucleus in here, these are some mitochondria in there, and that's that. So these are my rods, these are my cones. This is my rhodopsin. So let's talk about how these work real fast. The first thing I want to do is I want to talk about rhodopsin. So what I'm going to do is where you see these little lines in here, that's actually a disc. And the disc has different parts. And you can see, I would actually have 3 discs that are right here. This should be my rhodopsin right here. Okay, there's my rhodopsin right there. So if I were to pull this out, it would look like this here. Now, this part would be called opsin, and this would be called retinal. Okay, so my rhodopsin is made up of retinal, and it's made up of opsin. Okay, it's made up of retinal, it's made up of opsin. Retinal is actually made of vitamin A. Here's what's going to happen. When I am going to have a light ray hit the rhodopsin, this right now, this retinal, is basically what we call 11-cis retinal. This is going to straighten out. So let me find my red right here. This is going to straighten out. So now I go like this. That is going to straighten out. So now it's going to become straight like this. Okay, let me see if I got my brown. There's my brown. So it's just going to become straight. So it's kind of like this. Imagine my arms like this, right? This is the bent part here. Light hits it and now kind of flops over. When that happens, we are going to be calling this all trans. When this becomes all trans, it's going to separate from the rhodopsin. Okay, and then this is going to separate. So now I have my retinal down here. And remember, it's all trans retinal. And I have my opsin. Still right here. Now eventually what's going to happen is I am going to have ATP and an enzyme, and it's going to change this back so that it's bent again. When it oh, by the way, when this separates like this is called bleaching. When that separates, it's called bleaching. Okay, so if we can get down there. So when this separates, that's called bleaching. Now what's going to happen is eventually this is going to come back together. Okay, and I use brown instead of red. So just to stay consistent, Let's make this red again. Okay, and now I'm back to having rhodopsin there. Okay, so once again, down in here, I know it's hard to see, you can't see it right now, but let's just go like this. I'm gonna have my ATP become ADP. There's an enzyme that's gonna play a role in here. My retinol is gonna go from all trans back to cis trans, and then it's gonna join back into the rhodopsin. Or back to the opsin to make rhodopsin, okay? So now, let's take a look at the effect that this is gonna have. When we have opsin, opsin is basically going to play a role, and what it's going to do is it's going to have some steps, and eventually what's going to happen, this is when there's light. So and there's just a little bit of light. Eventually what's going to happen is the opsin is going to play a role when there's light. When there's no light, this does not happen. And that's going to send a signal to the brain. So when there's light, this occurs, and it sends a signal to the brain. When there's no light, this does not occur, the bleaching does not occur, and therefore no signal sent to the brain. So let's go like this. When there's light, I make opsin, and I'm going to go into what the role of opsin is in just a minute. And eventually I'm going to get a signal sent to the brain, and you can help see in low light. Okay? When there's no light, no opsin is made. So that bleaching that's going on here does not occur. When there's no opsin, no signal sent to the brain. And therefore, you don't see. Okay? Now, if you're in a high school class, that's all you need to know. All right, but let's continue on now if you are taking a more advanced class about what's going to happen. So first things first, let's come over here. And what I want to do is I am going to draw basically the inside of this disc. Okay, I'm just going to draw it as a square. And now, we're going to talk about first thing if there's no light. So we have a little protein here. And in this protein, what it's going to do is it's going to take something called GTP and turn it into cyclic GMP. We also have what cyclic GMP is going to do is it is going to open up channels. Okay, so this is going to basically cause these channels to open. These channels, we're going to have sodium and calcium enter. So sodium and calcium are going to enter. So let's just go right up in here. I get the cyclic GMP, and it's going to open up these channels. It's going to combine with this. It's going to open up sodium and calcium channels. So sodium and calcium are going to go in. When sodium and calcium go in, this becomes more positive. When this becomes more positive, this depolarizes. It's not an action potential, it's just depolarized. It's becoming more positive. When this becomes more positive, an electrical signal comes down the rod, okay? So when we get to the end here, to the synaptic terminal, I'm also gonna have more ion channels here. What's gonna happen is they're gonna open up and calcium's going to go in. When calcium goes in, what's gonna happen is we are going to release something called glutamate. When we release glutamate, what's going to happen is we are going to, usually glutamate will excite something, right? This is my bipolar cell here. In this case, my glutamate's going to come across and it's going to inhibit this bipolar cell. It does something that's called an IPSP. It's an inhibitory postsynaptic potential. What this means is that this is not going to be excited. This is actually going to be told to calm down. The IPSP is going to calm this down. When this gets calmed down, the inside becomes more negative and we're going to get hyperpolarization. Now, when we get hyperpolarization, we also have calcium channels that are down at this end of my bipolar cell. And what's going to happen is those are not going to open. So there's my calcium. Those are not going to open, so calcium is not going to enter. When calcium doesn't enter, I'm supposed to have glutamate that's released here, right? There should be glutamate here. And what happens in this case, the calcium channels don't open, the glutamate is not released. When the glutamate is not released, over here, when the glutamate is released, which we'll see in a minute, it should excite this, what we call an EPSP. In this case, I don't have the EPSP because of the fact that I didn't get the calcium being released. When the calcium's not released and this does not get excited, there's no action potential. There's no action potential. When there's no action potential, right, we're going to call this AP. When there's no action potential, no signal is sent to the brain. No signal sent to the brain, you can't see. You're in the dark, right? So there's no signal sent to the brain in this case, right? Let me see if I can move that just a little bit. No, I got that glare right there. So I'm going to just rewrite this down here. No signal sent to brain, okay? And this is all if there's no light. If there's no light, okay? So let's go back and take a look real quick. I have my GTP over here. I have my GTP over here, becomes cyclic GMP. When this becomes cyclic GMP, it's going to bond to my ion channels. When it bonds to the ion channels, the sodium and calcium are going to enter into here, causing this to become more positive. When that becomes more positive, it depolarizes, so it sends electricity down here. This is not an action potential. It's just sending electricity down here. That causes this calcium ion gate to open. Calcium goes in, it releases glutamate. The glutamate is actually going to calm this guy down. When it calms this guy down, we call that an IPSP, an inhibitory postsynaptic terminal. This is my synapse. This is postsynaptic, right? It's going to inhibit this. It's going to make it relax. Then what's going to happen is this is going to become more negative. It's going to be hyperpolarization. It doesn't have enough energy to open up this calcium channel. So calcium does not go in. Calcium does not go in, so glutamate is not released. When glutamate is not released, I cannot get this excited enough to cause an action potential. When there's no action potential, no signal is sent to the brain. Now, let's go back over here and look to see if there is light. So if you recall, what we said is we have something called opsin. All right, so I'm going to draw my little opsin molecule right here. Let's just draw it right here. No, let's draw it on this side. So I'm going to have my opsin, okay? Now, my light hits this, right? My retinal has become separated, so I'm going to draw my little retinal right there. That's my opsin molecule right here. By my opsin molecule, I have another molecule, a type of protein called a G protein. This is called transduction. And then I'm going to have something called, let me use orange here. I'm going to have another protein, actually an enzyme, well, which is a protein. And we're going to call this guy PDE. Okay, now I'm drawing these as if they're all separate. Remember, we have these going all around our cell membrane. This is the inside of the disc. So I'm talking about as if I'm talking about one of these green things here, okay? Now we're talking about, let me just write this up here. This is when there's no light. Okay, that's when there's no light. So now let's talk about what's going to happen here. I have my opsin, right? The light has hit it. We've had that whole process I showed you a minute ago in which we have the retinal separated from this called bleaching. This opsin is going to activate this transducin. So this comes over and it activates that. When this gets activated, what's going to happen is it's going to activate PDE. Now remember, I have this stuff all around my cell membrane, right? What PDE is going to do is it's going to separate my cyclic GMP. So my cyclic GMP just becomes GMP. Okay, so now it's just GMP. So what effect is that going to have now? So let's take a look. If we go back over here, we're going back and forth a lot here today. So what's going to happen? Now this is if there is light. So now I have my GMP, right? Not cyclic GMP, I just have GMP. What's going to happen now is this is not going to be able to bond to my sodium and calcium gates or my ion channels. this is not going to happen. Because that doesn't happen, this gate remains closed. Because this gate remains closed, my sodium and my calcium cannot enter. Because my sodium and sodium and calcium cannot enter, the inside becomes more negative. We are going to call this hyperpolarization. It becomes more negative, right? So the inside becomes more negative, but now that's hyperpolarization. Because the inside has become more negative, we don't have enough energy to open up this calcium gate down here, right? So this is not going to enter, right? This gate stays closed. Because that gate stays closed now, my glutamate is not released. So my glutamate is going to stay inside here. My glutamate is not released. Because my glutamate is not released, this bipolar cell now is not inhibited. It's not inhibited. The bipolar cell is not inhibited. Because the bipolar cell is not inhibited, what's going to happen is this is actually going to be excited, right? And it's going to depolarize. Remember, this is not an action potential. It's just becoming more positive. But because it becomes more positive, what's going to happen is we are going to open up this, the calcium, right? The calcium gates are going to open. Sorry, I lost my train of thought there for a second. Calcium gates are going to open and calcium's going to go in. When calcium goes in, now my glutamate's released, right, into this ganglion cell. When the glutamate is released into that ganglion cell, what's going to happen is I am going to get something called an EPSP, right? That's going to excite the ganglion cell, right? So the ganglion cell now becomes excited. Because the ganglion cell becomes excited, what's going to happen now is I am going to get an increase in basically electricity in there, right? And I'm going to get an action potential. Now, because I have an action potential, we can send a signal to the brain, and I see at night. As I send a signal to the brain, and I see at night. Remember, this is the optic nerve right here, so this is going to the brain. So let's go through all of this one more time. Okay, let's go all the way back over here. This is what happens if there's light. So I'm going to put light down in here. If there's light, up here there's no light. So if there's no light, my GTP becomes cyclic GMP. My cyclic GMP is going to bond onto calcium, onto sodium and calcium ion channels, causing them to open. Sodium and calcium are going to go in. Inside becomes more positive. As that becomes more positive, it depolarizes, so it's going to send electricity down here. As that happens, this is going to open the calcium gate. When the calcium gate opens, we're going to release glutamate. Glutamate's going to come across and it's going to go into this bipolar cell. When it goes into that bipolar cell, what's going to happen is this is going to activate. I'm sorry, it's going to inhibit it. It's going to inhibit it. When it inhibits it, now my calcium gate over here can't open, and because that can't open, I can't release glutamate. When I can't release glutamate, I can't get this excited enough to send an action potential to the brain, so no signal is sent to the brain. That's if there's no light. If there is light, let's go back over here. This is a lot of going back and forth. What's going to happen is my GTP is going to become cyclic GMP, but because my opsin has gone through that process of bleaching, it's going to activate something called transducin. The transducin is going to activate my polydiesterase, and my polydiesterase is basically going to break down the cyclic GMP into just GMP. When it becomes GMP, I should go like this, right? When it becomes GMP, it cannot bond to the ion gate. When it can't bond to this ion channel or ion gate, my sodium and calcium cannot go in. When my sodium and calcium cannot go in, it becomes more negative. That's called hyperpolarization. Basically very few signals are sent down here. Because of that, my calcium gate does not open. When my calcium gate does not open, I cannot release glutamate. When I cannot release glutamate, I cannot calm this bipolar cell down. When I can't calm that bipolar cell down, there's more electricity, there's more energy to open up this calcium gate. The calcium gate opens, releases glutamate. Whoa, I didn't write glutamate. So now I'm going to have glutamate, and the glutamate is going to cause an EPSP. By the way, EPSP is an excitatory postsynaptic potential, right? So here's my synapse. I'm postsynapse again, right? Over here I'm saying the synapse was over here. Oh, now we're here, we're postsynaptic. This becomes excited. Because it becomes excited, sends an action potential to the brain, okay? So that's the way that's gonna work. Now, really quick, and I'm not gonna spend too much time on this, you do have your horizontal cells that are going to be in here, okay? And what's gonna happen is there's actually, in this case here, when glutamate's released, what it's gonna do is it's gonna, and I know I draw it up and down, but imagine it's coming in and out. We're just looking at one little row. You have millions and millions of these, right? What's gonna happen is the glutamate will actually activate the horizontal cell. And when it does that, what the horizontal cell is gonna do is it's gonna release a neurotransmitter called GABA, and the neurotransmitter is going to basically inhibit action potentials going to the brain. You have the same thing over here. Your amacrine cells are more likely to be over here. And this is going to release several different types of neurotransmitters, which are also going to stop the release of signals going to the brain. So that's it for the rods and the cones. I hope you enjoyed the video. It's a lot. It's confusing. One's turning on, the other's turning off. But anyways, thank you so much for watching. Go Kings. If you like the video, please hit the like and subscribe button, and we will see you next time. Take care.